Hello again. This is a very long chapter. This is only the second part out of three. Chapter 21, the immune system, the innate and adaptive body defenses. Welcome back. So, I pretty much like hinted out that our body defenses is made up of innate and adaptive defenses. Innate means you're born with it. So these will be your surface barriers, your skin, your mucous membranes. And you got that on the first video. The internal defenses, which will be uh, it will be like starting whenever the microorganism would have the chance to breach um, the first part of the innate defenses. You have a cut, the microorganism got in, then your second defense will pretty much like kick in. And these internal defenses will be our fever, inflammation, phagocytes, antimicrobial proteins, and the natural killer cells. Now, if these defenses again will be breached and everything, the microorganisms would escape into the bloodstream, the adaptive defenses will start to kick in. This is now uh, this is now learning. This particular video is going to teach us how these adaptive defenses would come into play. And if I'm not mistaken, part also of the internal defenses will be discussed also. So to continue, the adaptive defenses will be defenses against specific pathogens that change body cells. And they are adaptive because our body, our body cells, our immune system cells have not been exposed to it, and therefore we adapt to them. Okay, we need exposure in order to adapt to them. Pretty much, this is unlike the innate where we're actually born with it, but together the adaptive plus the innate will be our total defense against injuries and pathogens. This adaptive immune system is a specific defense system. It eliminates almost any pathogen or abnormal cell in the body. Now, certain activities would the certain activities of this defense mechanism would amplify the inflammatory response. It would also be activating complement system, but there's just something, a shortcoming. This is not something that is automatic or automatically done. It needs priming. We need an initial exposure to the specific foreign substance. And this priming takes time. So let's take a look at the characteristics of adaptive immunity. Well, the adaptive immunity is specific. It recognizes the target's specific antigen. It is systemic. Keep on pointing to it without my pointer. Like it is systemic, and this means it is not restricted to the initial site. It has memory. It mounts an even stronger attack to known antigens. This means the second or the subsequent exposure pretty much would be mounting an even stronger attack. And there are two main branches of this adaptive defense. The first one is what you call the humoral or antibody mediated immunity. And the second one is your cellular or the cell mediated immunity. Humoral. Humor means blood. And so the humoral mediated immunity have would be producing something that is directed or that is actually going to be released in the bloodstream. And this is actually the antibody mediated immunity. The cellular mediated immunity. is actually when our white blood cells would be like real soldiers in combat. That's actually going to be doing one-on-one kill of the um, of the pathogen. The pathogen that would have the foreign antigen will be recognized, and they will go after the pathogen for a kill. The humoral immunity produces antibodies. It is actually done by the lymphocytes. They circulate freely in body fluids. They bind temporarily to the target cells, and then they temporarily inactivate. They're going to be temporarily inactivated. They are marked for destruction by phagocytes or the complement system. The humoral immunity has extracellular targets. They need to recognize and be able to target components of the microorganism that is actually extracellular, and you call this the antigenic markers. Call them antigens. You could see the antigenic receptors right there, okay, of your B cell. Your B cell needs to recognize that particular antigen um, of the bacteria. Here, this is your bacteria. This bacteria will have a particular antigen that it needs to recognize in order that it can actually kill any organism or anything that would carry the antigen. Lymphocytes act against the target cells two ways. Number one, either directly by killing the infected cells itself, or indirectly by releasing chemicals that would enhance the inflammatory response or gather or pretty much like activate other lymphocytes or macrophages, or would affect the gathering or agglutination effect of the microorganism. Cellular immunity has cellular targets, meaning this is actually your cell mediated immunity. The target is actually the cell. Unlike the humoral, where the end effect should actually be released in the blood, in the cellular immunity, the target is the actual cell right there. So let's take a look at how this thing works. First thing we need to do is to know the word antigen. What are antigens? The antigens are substances that can mobilize adaptive defenses and provoke an immune response. A foreign substance that can get into your body that will not provoke an immune response is not an antigen. The one that would provoke an immune response will be an antigen. These antigens are targets of the adaptive immune responses. Most of them are large, complex molecules that is not normally found in the body, in our own body. You find the antigens outside, which means they belong to microorganisms. They are not self. So characteristics of antigens will be either complete antigen, which are actually usually large molecules, or haptens. They are incomplete. And pretty much a haptens most of the time will not be provoking an immune response. It will not elicit an immune response because it cannot be recognized because of its small size. However, over time, haptens can over time elicit an immune response if over time when it keeps on coming and getting into our system, when our proteins would chelate or would attach themselves to the haptens, then the haptens will now be recognized by our body as something foreign. Hence, these are examples of uh, allergies. When I first came to America, I did not have hay fever. I did not have any reaction to anything in the environment. After 5 years, and it's really like a clockwork, after 5 years of being in America, I started sneezing whenever there's actually a lot of pollen from the, from the tree, or whenever there's actually like a grass growing in the field during summer, during springtime. I started sneezing. It's because my haptens, or rather the haptens, or the antigenic markers of this grass are actually haptens. They're too small, and it takes time for people really to develop an allergy to them. It is some people don't even get the chance to have that until they're probably old. I got it after 5 years of being in the United States. There's no such thing as that kind of an allergy um, where I was born. But when I came to America, I got it. So, what is the difference between a regular molecule and an antigenic molecule? Well, an antigenic molecule, whether they are haptens or complete, contain what you call antigenic determinants. And antigens are actually substances or molecules that can provoke an immune response. Do we have self antigen? Do we have, are all antigens foreign? Don't we have antigens ourselves? Yeah, we do have our antigen that we are born with. You remember our antigenic markers, the antigen A, antigen B, the Rh antigen. These are the antigens of our blood. Those are the first antigenic markers that you have learned. We have. So, do we have any other antigenic markers? Yes, or epitopes? Yes, we do have them. We have them in our cells, and these are the characters of our cells. That's why organ transplant is not easy, because you have to consider all of the antigenic markers of each individual, which may not be compatible. And if they're not compatible, then you cannot mix them, which means that the antigenic markers will be recognized by the recipient's white blood cells, and they're going to be attacked. So we do have antigens. So complete antigens pretty much have two important functional properties. You need this property in order to become an effective antigen. The first one will be immunogenicity. Immunogenic. It has to be immunogenic. Immunogenicity is the ability to stimulate uh, proliferation of specific lymphocytes. And then the next one is reactivity. It's the ability to react with activated lymphocytes and antibodies released by the immunogenic reactions. Examples will be foreign proteins, the polysaccharides, lipids, nucleic acids. They are seen on many foreign invaders such as pollen and microorganisms. Incomplete antigens or haptens are small. They involve molecules that are really too small to be seen and so are not immunogenic by themselves. Examples of these will be your small peptides and nucleotides and some hormones. They may become immunogenic if haptens attaches to our own body protein, as I was explaining. Combination of a protein and a hapten is then seen as foreign, and then you're going to be developing an immunogenic reaction to it. It causes the immune system to mount an attack that is harmful to the person because it may attack a self protein as well as the hapten itself. So it can attack an autoimmune response because you're attacking your own protein that is attached already to the hapten. Example of this would be your allergies to poison ivy, to danders of animals, detergents, cosmetics, grass cuttings, pollen. So let's take a look at what we mean by antigenic determinants. These are the antigenic determinants are epitopes, not epitaphs, all right? Epitopes. An epitaph is actually something that you put on the grave site. Nope, that's not what we meant. We mean epitopes. These are small portions of antigens. They are antigenic determinants. They are parts of the antigen that antibodies or lymphocyte would need to bind to. So, would they just simply binding them when they see them? No. Like this one here is a rounded epitope. That particular antibody that was produced by a lymphocyte would need to have a rounded structure in order to pretty much like attach to it. If it will have a triangular or a squarish attachment, it is not going to be um, effective in attaching itself to this epitope. So an antigen will attach to an antibody, rather this antibody that's been produced by a white blood cell will uh, will have to react to an antigenic determinant, and we should know the shape of the antigenic determinant so that it can effectively incapacitate the bearer of the antigenic determinant. If this antigen or belongs to a microorganism and this is actually the one that's making us sick, then this needs to be like discarded. And you cannot just simply discard them in a flowing, fast flowing blood. You need to make sure that you disable them first in their movements, precipitate them or slow them down so that the macrophages can go after them and digest them. So there is actually a big job to be doing this particular work. It is not only recognizing the antigenic determinants, it is producing the right antibodies so that the antigenic determinants can actually latch onto it and to be able to present it really to a macrophage in order that it can be digested. Self antigens. All cells are covered with a variety of protein located on surface. And these are not antigenic to us. These are our proteins and therefore we should not be reacting to that. But it may be antigenic to others. That's why we have to be very careful that there's going to be good matching, cross matching before a transfusion or a organ transplant is going to happen. One set of important self protein is actually a group of glycoprotein, which you call the MHC. MHC stands for major histocompatibility. So, major histocompatibility complex 1 and 2. So, there are two types of major histocompatibility complex. And you can see already the two major histocompatibility complex anatomically how different they are. These protein or markers, when they are in the plasma membrane, this is the plasma membrane, you have in the MHC1, in the class 1, you have only one transmembrane protein that anchors itself onto the plasma membrane. In the class 2, you have two transmembrane protein that is anchored to the plasma membrane. Both of them will have peptide binding cleft, and they're made up of pretty much like proteins, globular proteins, and both of them contain group, and the lymphocytes, the T lymphocytes, will only recognize antigens, meaning foreign antigens, that are presented to them by the major histocompatibility complex. So this means that the foreign antigens will have to be placed right there so that it can be recognized by T lymphocytes. Wow, that is actually different. That's actually difficult. How do you do that? Well, there are ways of doing that. That's why we have thousands of lymphocytes that are working in our favor. So, the class 1 major histocompatibility complex proteins. They're present in membranes of all nucleated cells. All cells that have nucleus will have the MHC1, which means our red blood cells, because it is not nucleated, will not have the MHC1 protein. The MHC, the class 2, the MHC2 protein, is present only in membranes of antigen presenting cells. I ask you to remember the antigen presenting cells because these are the heroes of our story in our defense. And soon enough, I guess in this particular video, I am going to explain to you why they become or how they become heroes. Examples of antigen presenters will be your monocytes, your macrophages, your dendritic cells. And these MHC2 proteins appear only when the cell is processing an antigen. Okay, so you have your types of lymphocytes here. You have your B cell, which will become as they become well, a naive lymphocyte have not seen an antigen. Once they are presented the antigen and they become reactive, they become plasma cell. They will start cloning themselves. They will go to the bloodstream and become plasma cells. And they're as see, if there's going to be one plasma cell that is residing in the in the lymphoid organ, in the lymph node particularly, that one particular B cell will be so happy that he was able to recognize one particular antigen. He's going to be happy. He said, I saw one and I was presented one, and now I can become a true soldier. I have to go out and kill all of the all of the microorganism that would bear this antigen. It is now in my memory. I recognize it. All right. So when it goes out, and there's actually a lot of those microorganisms there, he cannot actually like start killing all of them or start um, um, attacking them. No, not one lymphocyte. So what's going to happen first is that it will become a plasma cell, and this plasma cell will need to clone itself. Once it has cloned itself, then they will all lots of them will they will all be producing the antibody, and this antibody is going to be neutralizing the effect of the antigen. You have the other side, which is actually the T cell. These are your B cells. They produce antibody. The T cells are actually different. The work of the T cells are actually different. You have two types of T cells. You have T cells that would have a protein on it, which you call a protein co-receptor, which you call a CD4. This actually becomes the helper T cell. The CD4 T cells become helper T cells. The other type of T cell has a co-receptor that is called the CD8. This is actually the one that becomes the cytotoxic T cell. When do they become? Well, if they are naive and they have not really encountered any antigens yet, then you call them just a regular CD4 T cell or a CD8 T cell. But once a CD4 T cell recognizes an antigen or was presented an antigen and would recognize it, then it is going to be it's only going to become a helper T cell. It will now do the job of a helper T cell. The CD8, once it becomes activated, will now become a killer T cell. It will now do the job of a killer T cell, which is really not easy. The job of the helper and the cytotoxic or the killer T cell is actually as important as the job of the lymphocyte B, that is actually producing antibodies. They're all very important cells to the adaptive immunity, but they have different functions. Ooh, we now go to my favorite cell. I told you that these are this cell here is a very special cell because it's a hero. Now think of it as this way. This cell here, this is how it goes, and this is how it becomes a hero. This cell here would actually like engulf, usually a macrophage cell. It will engulf uh, it will engulf a pathogen. This is actually your bacteria. It has all the antigenic markers of the bacteria right there. It is now going to be swallowed by this big macrophage, and then the macrophage is now going to digest it, and once it is digested, all of those small units of antigenic markers pretty much will be inside the cell and would be released outside right here as what do you call this as antigenic markers. So what it's going to do is that it's going inside it. It's going to produce some sort of uh, a container to hold on to these antigenic markers, to these epitopes, and would release it, not really release in the bloodstream, but it would actually uh, it will actually present it in its plasma membrane. You could see it that way. It is still connected to the bacteria and it is presented there. So now this guy here, this big guy here, can now sit in one corner and kind of like saying, "Hey guys, look at me now. I have the antigen that is actually on my plasma membrane. Come and get me." Which means come and study, present, if you want presented antigen, if you would like to see how the antigen look like, come and see it because I have plenty on my skin. So all of those naive white blood cells or lymphocytes will now be going to this antigen presenter and would feel the antigenic marker, would recognize and would study how the shape of the antigenic marker goes. Quite neat, huh? But then how do they become heroes? Well, they become heroes because of that. They also could be killed because of that. They are actually carrying antigens, antigenic markers in their own plasma membrane. And if there's going to be already an active killer T cell, a lymphocyte will just be producing antibody against that. But if you have a killer T cell roaming around and would see that antigenic marker on its skin, it could go after that antigen presenter and would kill that antigen presenter. But then before it dies, it has already done its purpose. It has already presented the antigen to a lot of lymphocytes. It is really very hard to find an antigen in a rush of blood. See how the blood is actually like flowing so fast, and you have a lot of white blood cells and microorganisms. It's very difficult. You need to stay still so that the antigenic marker can latch into the protein of a lymphocyte and pretty much recognize it as a foreign antigen. It is not easy to be doing. That's why it takes time. Priming. This is actually what you call priming. Priming an antigen takes time because it is not easy, because all of these are actually moving inside our bloodstream. They have to go after those microorganisms. They have to present the antigen so that it can actually be recognized. It takes time. And your antigen presenters are not only the macrophages, most of them are the macrophages, the monocytes, and the neutrophil. A lot of them, though, are what you call dendritic cells because dendritic cells are immune cells that process antigen material that are present in the skin. These are the Langerhans cells of the skin, and they are found on the skin right there in the epidermis of the skin, the basal layer of the epidermis of the skin, and they're also found in the lining of the nose, the lungs, the stomach, and the intestines. Sometimes a dendritic cell presents on the surface of other cells to induce an immune response. Thus, the functioning pretty much as an antigen presenting, as an anti, thus they become an antigen presenting cell. Macrophages also function as, here you go, as antigen presenting cells. Before activation and differentiation, B cells can also function as an antigen presenting cell. The macrophages are wandering. Some of them are fixed, though, like the Kupffer cells in the liver, the mesangial cells of the kidney, the microglial cells of the brain, the osteoclast of the bone, the alveolar and peritoneal macrophages. These are all fixed in their respective organs. The B cells do not activate naive T cells. They present antigens to helper T cells to assist in their own activation. So, pretty much, let's take a look at uh, lymphocytes. Let's take a look at the development, the maturation, and the activation. How do they become soldiers, the lymphocytes? Well, we have two types of lymphocyte. One is your T cell, the other one is your B cell. They're all going to be born coming from a hemocytoblast. The great great grandmother of all blood cells will be your hemocytoblast. They all come from the hemocytoblast, and one becomes the thymus, the T cell, and would travel to the thymus and would mature there. The B cells would stay where they were actually born in the red bone marrow and pretty much uh, become immunocompetent, developed and immunocompetent in the bone marrow. The B and the T lymphocytes share a common development from here and pretty much would split out in two different organs in the secondary organ once they start their immunocompetency. And so they mature. The lymphocyte precursor that is actually destined to become T cell would migrate in the blood to the thymus and mature there. The B cells in the in the bone marrow stays in the bone marrow, and during the maturation process would develop immunocompetency and self-tolerance in the bone marrow. When do they go out? Do they stay there? Or do they go somewhere after they actually develop? Uh, no, they don't stay there. Stay there. They go outside and they will be seeding secondary lymphoid organs and would go in circulation. They're immunocompetent, but would still be naive because they have not been presented any antigen. When they leave the bone marrow and the thymus, they seed the secondary lymphoid organs and circulate through the blood and the lymph. So once they are already, let's say there's actually a diagram here of the lymph. Once they are in the lymph and they encounter, this is actually the most likely place, the place of the greatest a chance that they are going to be encountering an antigen is actually your lymph node. So the lymph node is pretty much filtering the lymph, which is actually the fluid that they get from the extremities and all over the body, most of the time the antigen encounter and activation happens right there in the lymph node, where a lymphocyte's antigen receptors bind to its antigen, then the lymphocyte is already activated. So naive lymphocytes first encounter with the antigen would trigger it to develop, and lymphocyte that is selected will differentiate into active cell by binding its specific antigen and then cloning itself. You have only one lymphocyte, it has to clone itself. Okay, if the correct signals are present, if they are co-stimulated, the lymphocyte will complete its differentiation and will become an active cell. Proliferation and differentiation. The active or the activated lymphocyte will now multiply, will now clone itself, and then differentiate into effector cells and memory cells. What's the difference between an effector and a memory cell? An effector cell will go into the plasma and will become plasma cells and produce antibodies. Meanwhile, the memory cells will stay in the hub and would just be there, and because they have the memory of the antigen, they are not supposed to go anywhere else close to the battlefield because they don't want to get killed. They are not going to be killed because they have the memory. You need the memory for the second time that we encounter that, the memory cells will have to do their work. Let's take a look at the antigen receptor diversity. The genes, not the antigens, would determine which foreign substance the immune system will recognize. A variety of immune cell receptors are result of acquired genetic knowledge of microbes. Throughout the years that humans have evolved, we have developed more than 25,000 different gene codes, and these codes for up to 1 billion different types of lymphocyte antigen receptors. We have a lot throughout the years that we have evolved here on earth as humans. The transfer of genes, our ancestors had transferred down to us the type of antigenic determinants that we have encountered or they have encountered. We carry it, the knowledge, we carry it in our genes, which humans have actually pretty much like developed over the years, millions of years of our evolution. And there's a huge variety of receptors, and gene segments are shuffled around. It's only 25,000 or 30,000 genes, but when they're shuffled around like in lotto, they result in many combinations and would account for about more than 1 billion different types of antigen receptors. So, how does the lymphocytes get to be educated with this billion? Okay, one lymphocyte, one antigen. Uh, you are hoping that the lymphocyte will carry the knowledge on how to um, um, kill this particular type of antigen and recognize it. How do they get educated? Well, first of all, the T cells mature in the thymus under what you call a negative and positive selection pressures. They're called tests. The positive selection process is where the is actually the one that selects the T cells that's capable of recognizing the self MHC proteins. And those that cannot recognize will be destroyed by apoptosis. So by positive selection process, they need to recognize the self proteins. You need to know what is self So you need to know that the self should not be killed. If the T cells cannot recognize the self, right there and there, they are going to be destroyed by apoptosis in the thymus gland. So right there, when the T cells get out or emerge out of the thymus gland, you know that they have already undergone the positive selection process. All of the white blood cells, the T cells, the killer T cells, and the would-be helper T cells that would emerge out of the thymus gland would already know self. They know that they should not kill self. Because if they will not recognize self right there in the thymus gland, they will already be killed. There is also another test which you call that's actually being done. We call them the negative selection process. This will prompt the apoptosis or the killing of T cells that bind to self antigen displayed by the self MHC. You are not supposed to, they are not supposed to bind onto self, because by binding onto self, then you recognize that as self and you can kill that self. You call this process clonal deletion. This will ensure self tolerance. Here you go, guys. In the positive selection, in the positive clonal selection, the T cells must recognize self major histocompatibility complex. This is the self major histocompatibility complex. You have to know that this is self. Recognizing self results in survival, which means you know that you're not supposed to kill self. You have to know which is self, because if you cannot recognize self, you might end up killing the self. In the negative selection, T cells must not recognize self antigen. If you happen to recognize self, that means you recognize it as an antigen, you can get killed by apoptosis. So you cannot actually recognize it. That's actually what you call negative selection. So guys, these are your overview of the B cells and the T cells. I'm hoping, guys, that you will uh, take a look at this. This is going to be a big help for your review. Okay, let's take a look at the clonal selection of a B cell. When B cells encounter target antigen, that's your target antigen right there, it provokes what you call a humoral immune response. Antigens, once it is actually established that that's an antigen and recognized that that is an antigen, antibodies that are specific for that particular antigen are then produced by these B cells. B cells are activated when antigens bind to surface receptors by cross-linking them, which then triggers receptor-mediated endocytosis of the cross-linkage antigen receptor complexes. You call this clonal selection, and they lead to proliferation and differentiation of B cells into the effector B cells. Most cloned cells, this is actually your cloned activated B cell, would actually become plasma cells. They're going to be cloned, and these plasma cells are the ones that are going to produce the antibodies. However, there could be like one particular cell that would not be responding to producing antibody, but would just be left behind in the lymph node not doing anything. But they are actually important because they carry the memory. They are called the memory B cells. Here's actually the clonal selection. This is actually your clones. Once they are activated, the clones will now be producing antibodies, lots of antibodies. So, how does our body remember the pathogens that we encounter? How do we remember them? Well, we remember them through the memory cells. The primary the cell in the primary immune response, the cell proliferation and differentiation upon exposure to the antigen for the first time is about 3 to 6 days. It will peak, the peak levels of the plasma antibody would be reached in 10 days. So it takes time. Then the antibody levels will decline. The white blood cell will continue to produce antibody until they feel that there's already enough antibody in the plasma. Then they will stop producing antibody. It will decline. This is actually the peak for the antibody production during the first exposure to the antigen A. In the second exposure, take a look at the graph. In the second exposure to the antigen A, it is actually faster, okay, and the production of antibody is much more than this. It's because of the memory cells. It doesn't take time anymore because there are already memory cells that know how to make the antibody, and the next thing that they're going to do will be just to produce massive amount of antibody. This is when the second exposure you don't really get sick much anymore. Probably will get sick a little bit, but not anymore much. So re-exposure to the same antigen gives faster and more prolonged, more effective response. Desensitized memory cells, which is our next heroes, provide immunological memory. The response is within hours, not days. The antibody peak will be in 2 to 3 days at a much higher level and can be maintained for weeks, even months. So, our humoral immunity, which is the production of antibody by the B cells, is now categorized into two. One is what you call active, and the other one is passive. In the active humoral immunity, the B cells encounter antigens and produce specific antibodies against them. And there are two types of active humoral immunity. The natural, the naturally acquired, which is actually formed in response to an actual bacterial or viral infection. You actually get sick here. Or artificially acquired, you got your uh, immunity through vaccine or dead attenuated pathogens, and they provide antigenic determinants that are immunogenic and reactive. They spare us the symptoms of the primary response. We don't get sick here because we are actually the exposure is already through a vaccine. On the passive immunity, this occurs when ready-made antibodies are introduced into the body. B cells are not challenged by antigens. Immunological memory does not occur. Protection ends when antibodies will degrade. And there are two types of humoral immunity. One is naturally acquired. So the antibodies are passed on to us from our mother, or pretty much like via the placenta, the mother's milk. And these are already natural antibodies. The one that's artificial is when you receive actual injections of exogenous immunoglobulin. So what does an antibody look like? Oh, That's it, guys. That's a molecule. It looks like a letter Y. That's a molecule of an antibody. And those arms are actually like the antigen binding site. These are the ones where the antigens will bind into. Okay, I will have to pause here because I need to go get some water. Antibodies, also known as immunoglobulins, are proteins secreted by plasma cells. They make up a gamma globulin portion of the blood, and they are capable of binding specifically with antigens detected by B cells. They are grouped into one of the five classes. Overall, they are actually either T-shaped or Y-shaped antibody monomer, and they're made up of four looping polypeptide chains linked by disulfide bonds. And so they're made up of and um, heavy chains and light chains. There are two identical heavy chains and two identical light chains. The variable region, the V, is actually where each arm combined to form two identical antigen binding sites. So this is your antigen binding sites on top. So there are five types of antibodies. You have the IgA secreted into the mucus, the saliva, the tears, the colostrum, and they tag pathogens for destruction. The IgA or the immunoglobulin D. These are B cell receptors. They stimulate the release of your IgM. There's one. IgE is actually the immunoglobulin that binds to mast cells and basophils. They're actually for allergy and antiparasitic activity. So your eosinophil actually will produce the IgE. IgG or immunoglobulin G will bind to phagocytes, the main blood antibody for secondary response. This is actually very important. And they can cross the placental barrier. The immunoglobulin M will fix the complement. It is the main antibody of primary responses. The B cell receptor is actually produced by B cells that would have the receptors for the M, and it is also part of the immune system memory. So, how do antibodies fight, or what is the mechanism of action of antibodies? Well, they do different things. They could precipitate, they could agglutinate, they could neutralize, they could pretty much like increase inflammation, and um, they can stimulate the complement reaction. In precipitation, this is exactly how those are antibodies that would be latching on to the red antigen. Pretty much the antigens, when they're just like free floating little dots, when the antibodies would uh, latch onto them, they become insoluble, they become heavier, and so they would slow down, they would aid in the elimination of the macrophages by the macrophages. And agglutination is when the antibodies are actually, or the antigens are found right there in the plasma membrane. These are what red blood cells, and these are the antibodies that agglutinate red blood cells during a mismatched transfusion. Neutralization, pretty much this would mask the dangerous part of a pathogen. When these antigenic markers are actually the dangerous part of the pathogen, the antibodies will latch onto them and the danger is actually neutralized. Inflammation, when antibodies would be latching onto the antigens and would elicit um, the formation or the release of inflammatory chemicals such as histamine, or it will increase the immune mobility, then that's actually already stimulating the inflammatory process. Or they can actually stimulate the complement system. Complement protein will be formed. This is actually your complement protein, the MAC, they call it the MAC, and will perforate the cell. And then the membrane will allow water to get into it, and it will cause the bacteria or the pathogen to explode. All other mechanism of action of antibodies will promote opsonization and phagocytosis. Opsonization means uh, an opsonin or it becomes like a substance that could agglutinate or clump together. And once they're clumped together, they become heavy and they can be um, eliminated by phagocytosis. This one here, when they activate the complement system, they do not promote phagocytosis, but they promote the actual bursting of the phagocyte, rather the bacteria itself. That's it, guys. We're now the end, and I will see you in the last video for the immune response, the video C. That's the end of B now.